Welcome to the another video of Unit 1. Last video of Unit 1. Let's get started. 1. Dispensers and auto-pipettes. Definition. Dispensers and auto-pipettes are laboratory instruments used for accurate and precise measurement and transfer of liquids in small volumes. Laboratory use. Used in biological, chemical, and medical labs for handling precise liquid volumes. Essential for sample preparation, reagent addition, and dilution procedures. VVI, very important information. Auto-pipettes come in fixed or variable volume types. Accuracy depends on proper handling and calibration. Must use appropriate pipette tips to prevent contamination. Avoid air bubbles to ensure precise liquid delivery. 2. Polymerase chain reaction, PCR. Definition. PCR is a technique used to amplify small DNA segments, making millions of copies for genetic analysis. Laboratory use. Used in forensic science, disease diagnostics, genetic research, and molecular biology studies. Helps detect pathogens like viruses and bacteria. VVI, very important information. Requires TIC polymerase, primers, nucleotides, and a thermal cycler. Follows 3 main steps: denaturation, annealing, and extension. Contamination can lead to false results. Always use sterile conditions. Real-time PCR, qPCR, allows quantitative DNA analysis. MCQs on dispensers and auto-pipettes. 1. What is the main function of an auto-pipette? A. Mixing liquids. B. Precise liquid transfer. C. Measuring solid samples. D. Heating solutions. Answer: B. Precise liquid transfer. Reason: auto-pipettes are designed to transfer specific volumes of liquid with high accuracy. 2. Which factor affects pipette accuracy the most? A. Temperature. B. Pipette tip size. C. User handling. D. Color of liquid. Answer: C. User handling. Reason: proper technique ensures correct volume measurement, while errors in handling can cause inaccuracy. 3. What is the best way to avoid contamination while pipetting? A. Using the same tip for all liquids. B. Disposing of tips after each use. C. Shaking the pipette. D. Using gloves only. Answer: B. Disposing of tips after each use. Reason: changing tips prevents cross-contamination between samples. 4. What should you do before dispensing liquid with a pipette? A. Warm up the pipette. B. Pre-wet the tip. C. Shake the liquid. D. Hold the pipette upside down. Answer: B. Pre-wet the tip. Reason: pre-wetting ensures consistent volume accuracy by preventing liquid adhesion inside the tip. 5. Which type of pipette is most commonly used in molecular biology? A. Serological pipette. B. Pasteur pipette. C. Micropipette. D. Graduated cylinder. Answer: C. Micropipette. Reason: micropipettes allow precise handling of small liquid volumes, essential for molecular biology experiments. 6. How can you calibrate an auto-pipette? A. By adjusting the plunger randomly. B. By checking with distilled water and a balance. C. By shaking the pipette. D. By using different colored liquids. Answer: B. By checking with distilled water and a balance. Reason: the known density of distilled water allows for precise calibration by weight measurement. 7. Which part of an auto-pipette ensures volume adjustment? A. Plunger. B. Tip ejector. C. Display screen. D. O-rings. Answer: A. Plunger. Reason: the plunger is responsible for controlling liquid uptake and dispensing in the pipette. 8. Why should pipettes be stored vertically? A. To look neat. B. To prevent liquid from entering the internal mechanism. C. To dry quickly. D. To keep them cold. Answer, B. To prevent liquid from entering the internal mechanism. Reason, improper storage can cause damage to internal parts, affecting accuracy. 9. Which liquid is commonly used to test pipette accuracy? A. Alcohol. B. Distilled water. C. Buffer solution. D. Acetone. Answer, B. Distilled water. Reason, distilled water has a known density, making it ideal for accuracy checks. 10. What should you never do when using a pipette? A. Hold it vertically. B. Pipette by mouth. C. Use disposable tips. D. Set the correct volume. Answer, B. Pipette by mouth. Reason, mouth pipetting is unsafe and can lead to contamination and health hazards. MCQs on PCR, polymerase chain reaction. 11. What is the purpose of PCR? A. Destroy DNA. B. Amplify DNA. C. Measure protein levels. D. Break down RNA. Answer, B. Amplify DNA. Reason, PCR is used to make multiple copies of a specific DNA sequence for analysis. 12. Which enzyme is used in PCR? A. DNA ligase. B. TUC polymerase. C. Restriction enzyme. D. RNA polymerase. Answer, B. TUC polymerase. Reason, TUC polymerase is heat resistant and can function at high temperatures during PCR cycling. 13. What is the first step in PCR? A. Annealing. B. Denaturation. C. Extension. D. Sequencing. Answer, B. Denaturation. Reason, denaturation separates the DNA strands to allow primer binding in the next step. 14. What is the typical temperature for DNA denaturation in PCR? A. 50 degrees Celsius. B. 72 degrees Celsius. C. 94 to 98 degrees Celsius. D. 37 degrees Celsius. Answer, C. 94 to 98 degrees Celsius. Reason, high temperatures are required to break hydrogen bonds between DNA strands. 15. Why are primers important in PCR? A. They degrade DNA. B. They start DNA synthesis. C. They remove contaminants. D. They act as enzymes. Answer, B. They start DNA synthesis. Reason, primers provide the starting point for DNA polymerase to add nucleotides. 16. Which component is not required for PCR? A. DNA template. B. RNA polymerase. C. Nucleotides. D. Primers. Answer, B. RNA polymerase. Reason, PCR uses DNA polymerase, TUC polymerase, not RNA polymerase, for DNA replication. 17. Which instrument is used to perform PCR? A. Centrifuge. B. Spectrophotometer. C. Thermal cycler. D. pH meter. Answer, C. Thermal cycler. Reason, a thermal cycler controls temperature changes for PCR cycling. 18. What is real-time PCR, qPCR, used for? A. Detecting RNA viruses. B. Measuring DNA amplification in real time. C. Studying proteins. D. Cloning bacteria. Answer, B. Measuring DNA amplification in real time. Reason, qPCR uses fluorescent dyes to track DNA amplification as it occurs. 19. What is a major source of error in PCR? A. Incorrect pipetting. B. Using fresh reagents. C. Proper thermal cycling. D. Using sterile tubes. Answer, A. Incorrect pipetting. Reason, small errors in pipetting can lead to incorrect DNA quantification and failed reactions. 20. What is a common contaminant in PCR reactions? A. RNA. B. DNA from previous samples. C. Salts. D. Proteins. Answer, B. DNA from previous samples. Reason, carryover DNA from past reactions can cause false positive results. If you like the video, then like, comment and share with your friends. Also don't forget to subscribe the channel. Thank you.